Maria Korbusch article in the microbiological journal. There, our study focuses on analyzing the influence of different concentrations of PM derived from the combustion of cottonwood and the medical masks on the growth, intensity, biofilm formation, capability, and antibiotic susceptibility of lactose-positive Escherichia coli. And why we pick up Escherichia coli? Because under normal physiological condition, E. coli ferments lactose and produces short-chain fatty acids. However, under the influence of external factors, E. coli can switch to proteolytic metabolism pathways, which are often implicated in the development of GI disorders and exhibit high resistance to therapeutic agents. So in our study, we found that particle matters in different doses. We use the 3 different doses, 18, 36, and 72 microgram of milliliters, significantly impact the growth curves of Escherichia coli. And also we found the number of viable cells after the incubation of particles matters with the Escherichia coli also decreased. But what we found next, and what was really very interesting, that particle matters which we got from the medical mask significantly impact antibiotic susceptibility of Escherichia coli. And we found that at concentration 18 and 36 microgram per ml, this strain tended to lose sensitivity to most antibiotics. And I seem that this results is really very interesting, maybe explain also the antibiotic resistance which we observed in current days, and these dates need to be continue to study. And I would say that this study, despite it was published in Microbiological Journal, because we need to defend my former PhD students and how the regulation from how the government affect the speed of publication. Unfortunately, we could not wait to publish in better journal, but I would say that this research, they are unique, and this research, it's only currently available research, the effect of PM on the separate bacteria. And here the conclusion from this research, and overall we think that the particles matters increased aggressiveness through reduced enzymatic activity, enhanced biofilm formation, and the emergence of resistance to ampicillin, ceftriaxone, and norfloxacin antibiotics. Okay, what is the next step? I tried to search for the next steps and translation from the bench to bedsides. And I found only one review article published in Environmental Pollution, which tried to summarize the current data on the probiotics, prebiotics, and symbiotics to prevent or combat air pollution consequences, the gut-lung axis. Nowadays, we know that it's not only gut-lung axis, it's also gut-brain axis, it's also gut-kidney axis, and so on. And according this data, we see that not enough data, especially proved in the clinical settings on the effect of different types of probiotics, prebiotics, and synbiotics to prevent the negative consequences of the air pollution. And I would say that this is the new avenue for the young researchers. And I would like to say thank you very much for my research group, and especially the Department of Immunology and Microbiology of the Institute of Biology and Medicine, which grew the list of great scientists who defends their PhD in interdisciplinary team of the histologists, microbiologists, immunologists, physiologists, and biochemists. Thank you very much.
Thank you for so amazing lecture. Dear colleagues, it's just the time to ask your questions. Please, do you have questions? Please.
Thank you very much.
Mm-hmm.
Thank you very much for a great talk. My question is actually, how do you differentiate the effects of the particulate matter that influence your microbiology testing from just a simple leachables that come from your material, etc.?
Okay, I didn't understand your questions. I understood English, but I didn't understand the context.
You mean some effects you see on growth rates, etc., can be exerted not by the particulate matter process, or actually the particles, but due to the leachables from such material, like such compounds that can be toxic to a culture, etc. So it's not the particulates which is matter. How do you differentiate these two effects?
Okay, so the particle matters, it's a solid compound, as you mentioned, which can consist of carbon, viruses, bacteria, heavy metals, and so on. But in our case, as I mentioned, we collaborated with the Institute of Biochemistry, and they produce that particle matters in the experimental, in laboratory condition, and they burn the piece of wood, and it didn't contain any other subject as a heavy metal and different types of aerosols which we have in real conditions. So this is the main question right now, and I really appreciate your question, so that we really have to separate different parts of particles matters which we have, because it's the composition, as you mentioned. And we study the effect of carbons, because we had the burning materials from the wood. And also as a continuation, and I didn't present these results, we continued our research, and we study, we compare the effect of particle matters as a result of burning of wood, different types of the wood, and also the heavy metals. And as you can also saw in my presentation, in the last article, we compare the medical masks and the wood, and we try to compare, like separate different parts of the particle matters which we which we meet in the air pollutants.
Okay, thank you.
Any question, please?
The significance of the air pollution and how to calculate this?
Very complex question, and I don't think that we really can separate the effect in real settings, that we can separate the effect, but in experimental settings, of course, we can separate this effect by comparison. And in our study, for example, then even in our study, we compare the particle matters from different types of the wood. And we found that particle matters from different type of the wood had no effect or less effect or more effect. In real life, and then we compare the effect of the particle matters from the burning of medical masks and the piece of wood, we found also then medical masks had more effect for development of the antibiotic resistances, but the wood didn't affect this feature of E. coli. So in the real everyday life, we of course can say that if the people live in the area with a high level of air pollution, of course, it can be the significant factor for the development of disease, besides other factor like genetics and maybe like type of lifestyles, diet, and so on. And in the rural area with the less level of air pollution, of course, this etiological factor will have the less impact in comparison to other factors like diet, lifestyles, genetics, and so on.
Thanks again, Hanna Mykolaievna, for your amazing answer your question.